cells, atoms, and bits, cutting-edge issues at the intersection of science, engineering, and ethics. I am Alan Jakimo, the moderator of the panel. Why don't I allow each of the participants to say a word about themselves, and then we'll continue.
Sure, I'm Jeanne Loring. I am a professor emeritus at Scripps Research. I've been working on human pluripotent stem cells for more than 20 years. And I founded a couple of companies, so I'm a scientist slash entrepreneur. And also I have other interests, including using stem cells to rescue endangered species. And I have a growing interest in discovering the effects of microgravity on stem cell development on the International Space Station.
Graham?
I'm Graham Parker. I'm on the research faculty at the Department of Pediatrics, Wayne State University, Detroit, Michigan, USA. As our gallant host mentioned, I'm also editor-in-chief of Stem Cells and Development and executive editor of Nucleic Acid Therapeutics. And probably largely because of my long involvement with the Stem Cell Journal, I'm also the ethical advisor for Mary Ann Leavitt Publishing. Thank you.
And Beth?
Hi, good afternoon. Beth Roxland. I'm an attorney and a bioethicist. I am an independent consultant as well as a practicing attorney. I've been working in the healthcare and research space for longer than I care to admit on film. Currently, I work as a litigator, as a healthcare lawyer, and in the regulatory space. Pertinent for today, which is not usually pertinent, is that in a prior life, I worked on pandemic preparedness and responses, everything from legal declarations of emergency to allocation of scarce resources in a pandemic. And it's become frightfully relevant, as everyone knows, in the past year plus, including even to the stem cell field. So I'm very excited for today.
So Beth, thank you, and your mentioning that work that you did on emergency preparedness and responses is a great segue into one of the major topics that we would like to cover today, and it's discussing and looking at exploring the effect that COVID-19, which the whole world has felt, the impact of a most surreal experience for everyone around the world on the scientific method. And there's a great example of the sort of thing that we're talking about in terms of looking at that impact that I'm going to ask Graham to begin with, and it involves the hydroxychloroquine episode that we all watched unfold over the course of the COVID-19 pandemic. So Graham, why don't I give you an opportunity to talk about that, and we'll go from there.
Well, thank you, Alan. First, I think it's worth pointing out that hydroxychloroquine is a very effective treatment. It just doesn't appear to be a very effective treatment for COVID-19. It's a very effective anti-malarial drug, and it's very effective in the treatment of rheumatoid arthritis and lupus. And the publications, plural, that Alan referring to, come from a very prestigious establishment in France that claimed that hydroxychloroquine would be effective. The interesting thing to note is that the first claims they were making was that it would limit the transmission of COVID-19. It would not necessarily achieve a treatment for COVID-19. But when other people started exploring in very small trials whether it was effective, they very quickly were saying, no, this does not actually help cure COVID-19. So that's the setting that we're setting this discussion against.
And Graham, we'll come back to that in a moment to just explore the whistleblowing aspects of that and the impact of whistleblowing on scientific method, and particularly the area of publication, not only of text, but also data sets. We'll come back to that in a moment. But before we do that, just furthering that concept that I think you just introduced in terms of how does the scientific method get applied in a COVID type of context. Jeanne, I think based on conversations that we've had, you pointed out that, and I'm going to refer to it as a relaxation for lack of a better word. If there's a better word, please correct me. But you pointed out that with respect to stem cell science and regenerative medicine, and the various organizations that are involved in that world, that during COVID, the scientific method may have been altered somewhat. So let me let you address that.
Yes, so as a veteran of many study sections to review grants for the NIH and a lot of other people, and having a lot of grants myself, I'm acutely aware of how a hot topic can come and take over and offer opportunities for science, which is perhaps not up to par. This has happened over and over and over again. But in this particular case, there were a lot of legitimate stem cell researchers who rose to the opportunity to get grant money to apply whatever it is they were doing to COVID-19. And quite a bit of work was done. It started a little late, and stem cells have not shown any utility for treating or even understanding COVID-19, with a few exceptions. People did study whether COVID-19 could infect cells in the brain, for example, and if so, what cells, but it was really taking advantage of a lot of money, which is what investigators always do. I mean, we can't do research without funding. And so when funding becomes available, it is perfectly understandable that people will morph their programs so that they are aligned with whatever it is that the government or some government wants to fund. I thought about doing it myself, and if I weren't so busy, I probably would have, because I thought there might be a niche somewhere that I could figure out just understanding the virus and still doing real science. But unfortunately, because the work was so rushed, most of what came out of it was frankly a waste of grant money. If there had been more time to carefully consider the validity of people's hypotheses and the expectations, all of this work was done as what we call, we lovingly call discovery research, which is funding for just finding out what happens if you do something. And that's fine. I think we got a body of results in that regard, but we did not come up with anything that was really relevant to human health. And then on the other side of the coin, with the stem cell clinics that everyone is aware of, certainly at this meeting, that have shown up all over the United States, in fact, all over the world, some up to maybe 1,000 of them we've heard. Many of them also took advantage of this. They had a list of diseases that they claimed that they could cure with fat cells, for example, autologous adipose stem cells. And they simply added COVID-19 to the list of diseases that they said they could treat. And that's just marketing. They wanted to catch the clicks on the internet that were looking for therapies for COVID-19. And I'm sure they gathered even more business for their clinics. And I just want to make it very clear, and the FDA has made it very clear, that there are no stem cell therapies that have any effect whatsoever on COVID-19, as well as a host of other diseases. And the only stem cell therapy that's currently approved by the FDA is the therapy for replacing bone marrow stem cells, hematopoietic stem cells, in cases of leukemia. So all the other aspects, all the other possibilities for the use of stem cells have not passed scrutiny, even in an EUA universe with the FDA. And I think it's just I just find this sort of thing to be a waste of resources. And I think Graham will talk more about how this generated an awful lot of publications of dubious value.
And thank you, Jeanne. And we do want to go delve into that point that you just made, referring to Graham. But before we do that, Beth, just going back to what you've done in the past regarding emergency preparedness and response, and looking at that from your perspective in terms of that work and the bioethics and keeping this, what Jeanne just referred to as kind of the EUA mindset, from your perspective, how does that all come together or fall apart?
I know, thank you for the question. Actually, what I find very interesting is that I spent many years working on altered standards of care during a pandemic. So we are in a horrible crisis situation. Our resources are short, our time is short, our abilities to do very much and to judge a situation are scarce. And how do we do the best in that situation in terms of, well, how do we change how we deliver healthcare, or how people make decisions for themselves or don't have the ability to make decisions for themselves in order to shift the focus to the potential for the greater good, because the greater population is what's facing mass tragedy. And that the shift has sort of I don't want to say it's a lower standard, because it's sort of a different goal of making the standard. So it actually applies to scientific protocols, apparently, as we've seen here, and to publications, as I see it. We have shifted our standards in a certain way. Some could call it lowering or loosening our standards for what we put forward, but with the justification that getting information out there or exploring certain scientific hypotheses in what Jeanne said very eloquently, if we had more time to consider the validity, and we're dealing though in situations where we have very little time to consider things. That's the whole idea of an emergency. How do we help the greater population either come forward with promising science or a promising publication to disseminate the information? And after a certain amount of time, though, as an ethicist, as a policymaker, I've been in sort of all shoes. There's a time, for better or for worse, in which you're still in a not ideal state, but you can take a second look at why you're applying these standards, and are these standards still appropriate? And look even back at what you've already done during the pandemic, as respects grant money or as respects publications that have come out, and take a second look at them, because we've got a bit more time now. And that part is actually almost more difficult than the very first part, where you're just scrambling and you're just doing your best. And so I very much look forward to this discussion, because again, I think this actually is both informed by other work and potentially inform other work. So
with that said, I think all particularly Jeanne's comments and Beth's comments, you've been circling this notion of, and again, I'm going to use a term which may not fit well, but a relaxation or an alteration of basic scientific method. And of course, in that context, that calls to mind the so-called gold standard of clinical studies for purposes of regulatory approval. And I just want to go back to Graham now, because I think that hydroxychloroquine example that he pointed to, I think that there's, as I understand it, there's some canonical whistleblowing things to talk about in the context of altered standards or different standards. So Graham, I just want to turn back to you to provide a little bit more color on that part of what you were talking about, that of course Jeanne and Beth also were addressing in their comments.
Thank you. I think it's first, maybe for a lot of the members of our audience, probably don't truly understand how science gets reviewed at a journal. I think there is a general feeling that, the work is handed from one ivory tower to another ivory tower, and then it's disseminated to the people. And I think it's worth recognizing just what an organic process it really is. A paper is submitted by someone who hopefully is trying to do good work, is trying to help the rest of the world, submits it to a journal. The editor then tries to find decent, qualified reviewers to review the paper. Then the editor has to review the reviews and make decisions concerning a publication. And what we faced with this pandemic is almost a perfect storm for bad science, because you have an incredible need combined with incredibly stretched resources and combined with an incredible knowledge vacuum, because all the best people to review any work concerning the pandemic are working on the pandemic. They don't have time to review papers. The best medical clinician researchers that you would want putting their eyes on any work that might get published are pulling 24-hour shifts, which is what they should be doing, and it's absolutely fit and right. But I just want to step back and make it clear that the majority of people that are trying to address COVID-19 are trying their very best. They aren't just opportunists. They're trying to help patients where there's a tremendous need. We had an interesting publication in Stem Cells and Development that used exosomes to treat patients actually in New Jersey. This wasn't happening in the third world. This was happening in a hospital. And the data are very promising. The exosomes actually did seem to improve the health of the patients. What was very interesting is what happened next after it was published. I got bombarded by emails from researchers who had done work similar to that, but in animal models before, and had never managed to get permission to try it in human patients. And I heard a very nice quote from someone associated with that work. He talked of their pharisaical approach to the publication, that he felt that these people either wanted to be able to do the work themselves and publish it, or that they weren't actually addressing the patient's needs, and therefore were walking on by on the other side and taking a holier than thou approach to any publication that was trying to relate it. My personal view, and believe me, a lot of people have very strong views about that work that was published, is that they did the work produced valid data, and it was worth sharing with the world. The world then has to make its decisions whether that was a treatment that was worth pursuing. But the hydroxychloroquine paper wasn't simply a case of somebody saying what they'd done and trying to do their best. It was work that was presenting data in such a way that I think doesn't fairly represent the work that was done. And so when someone sees that in a publication, the absolute right and proper thing to do is to contact the editor of that journal and say, look, this paper that was published, there are problems with the data. This is what Alan was referring to as whistleblowing. How the editor then responds to that then becomes a very interesting, obviously you want this, you don't want to be publishing retractions every single week, but you then have to communicate with the authors to try and find out what the truth was. And what happened with the hydroxychloroquine paper is that the researcher involved took a very, very reactive position to the complaints about their paper and have basically been targeting the whistleblower involved. And the almost frenzied support that this person has managed to garner in their attacking of this whistleblower has been absolutely horrifying to see, because we as editors absolutely rely on well-intentioned people letting us know when there's a problem with a publication. We need that. Science needs that. And the way the whistleblower has been treated has been nothing short of criminal.
Graham, can you just talk for 1 second for the folks who either are unfamiliar with it or unfamiliar with this specific issue with the hydroxychloroquine matter, what the background issue was with the paper itself, or with the publication process as it may be here, whichever aspect would be just sort of a introduction.
There's two main complaints about the paper. One has to do with revealing of possible conflicts of interests. Two is to do with not including data that should have been in the final analysis. I think that's the shortest way I can split up what the concerns are with this particular research.
So, go ahead, Beth. Go ahead.
Sorry, I think I'm usurping your role.
No, go ahead. Go ahead.
Wow.
That's fine.
No, because I love having such great experts that I get to ask the questions that are in my head.
Go ahead.
So for Graham, the two main issues that you point out, the conflicts of interest that may or may not have been there all along, and the lack of inclusion of data, rightfully, wrongfully, whatever. Could either of those be chalked up to what happens because it's a pandemic, because things were rushed? Or is this something more fundamental to processes, either on the scientific side that did the research or on the publication side?
Okay, so firstly, I'm very confident that whenever Jeanne Loring publishes a paper, she's very careful to make sure that the reviewers and the readers know what various pies she has her fingers in that might be related to how that piece of work is evaluated. The disclosure of conflicts of interest is absolutely sacrosanct, and it shouldn't matter whether you're in a pandemic, 40-day flood. You have to tell people what it is that you are going to benefit or not benefit from when you make a publication. That's the first thing to say. In terms of the data, if you can't trust yourself to report all of the data associated with a study, whether it be human trials or whether it be little rats in a cage getting treatment A or treatment B, then you should get out of science. The data have to be revealed, and it has to be all of the data. You can't just drop 6 subjects because that makes that the treatment effect was the same level as the control. And what happens in the paper that we're talking about was that 4 of the patients that received the treatment died. So yeah, funnily enough, they didn't contribute any more data to the study, but it's a pretty negative result to not include in your analysis. Okay, I'm making light of it. Obviously, it's not funny. People died, but that is basically the whistleblower we're talking about. I think we can talk about this candidly, because there's certainly not a private person. Is Elizabeth Fick, and she is very famous in the scientific community because she sends lots of emails to lots of editors saying, "Hey, I've looked at this paper. It's published in your journal. There's a problem with figure A. What are you going to do about it?" And you know what? You ignore someone like that at your peril, because she then sends it off to PubPeer, P-U-B-P-E-E-R. If you've got a quiet moment in your afternoon and you've never been there. It used to be Retraction Watch. We always used to talk about Retraction Watch, and I always used to say, never go to Retraction Watch, because if you're interested in science and you start reading that website, you're going to be there for hours. But PubPeer basically, I would say, focuses more on images and data issues than Retraction Watch, which seems to be more broader issues. I'm obviously speaking largely there. But yeah, PubPeer will just put up images from a paper and say, hey, look at this. Look at these Western blots are just basically a series of dots. Look at that. This was in paper A. Look at that. It was in paper B. It's the same Western blots. These people have produced the same image in 2 different papers. What are they going to do about it? And we need that, because an individual reviewer might miss it. I might look at a paper and I don't necessarily know whether that figure was in another paper or not. These people are doing us a service.
So with that said, Jeanne, your name came up a lot in that mini conversation that we just had. What are you, as a scientist, engineer, what are your thoughts? What are your thoughts listening to that and thinking about the publication process and the whistleblowing process? I'm sure you've got some thoughts there.
Yeah, well, personally, Elizabeth Bick is one of my heroes because I once encountered, I can see the phenomenon. We were doing a journal club once in my lab, and a picture was flashed up. It was from, I believe it was from Cell Stem Cell. It was not a minor journal. And I was looking at the picture that my graduate student had put up to, talk about that figure. And I noticed that there was a duplication in it. And I'm not an expert on this, but it seemed really obvious to me. And so I've done, and this has happened before. There have been times in which I've noticed something, and I do exactly what Graham said, which is I contact the editor. In this case, I knew the authors, and so I contacted the authors and said, I found this. Would you go ahead and tell the editor that you need to correct it? So I gave them a chance because I know them instead of going straight to the editor. But I also said at the very bottom, I said, if you don't do this, I will go to the editor, because I feel very strongly that we're all affected by this. All scientists are affected by the quality of science that's published. And I think we all know that there are really, really good papers that are innovative and have novel data and complete data and appropriate analysis. And I don't even know what the ratio is, but there seems like there's an awful lot of just junk that gets published. With the motivation, I think, and I think the motivation is probably really important. The motivation for me is that I want to share what I learned. There is an opportunity to share what you learned, and so I'm going to take advantage of it. But there is always an element, and there's certainly an element of the publish or perish concept, in which if you publish more papers than in the academic world, and even in biotech, if you publish more papers, then your reputation is greater. And that leads to, I don't know what it actually leads to. It hasn't led to anything for me yet, but maybe someday. But the idea is that that every paper that does such things so blatantly diminishes the rest of us. And I take it personally. I really do. And that's one of the reasons I think Elizabeth is such a hero to me, that she's gone right out there and she's looked for this stuff, and she's abandoned all the rest of the stuff that scientists do and decided this is going to be her calling. And I think I'm the occasional kind of whistleblower-ish person when I stumble on something, but I don't do it nearly as well as she does. And I think what she does is really important to science.
So that word that you just used, stumbling, let's put an asterisk on that, because I want to come back to that. But I want to circle the question that launched that dialogue that we just had was Beth's question about, hey, was the hydroxychloroquine situation something that was engendered by COVID, or is there a broader issue there? And I think listening to both you and Graham speak, I think the answer to Beth's question is it wasn't COVID engendered.
No, it was there already.
Exactly why I asked the question.
Yeah. Okay.
Yeah.
Especially with the conflicts of interest, to me, it seems that that's not, we started out the conversation with altered standards because of an emergency, but the aspects that were brought up that were problematic about whether it's the conflicts of interest or this is going just from Graham's distillation of it. I don't want to act like I've got firsthand expertise on all of this, but perhaps willful ignorance or non-transparency for deaths and whatnot. Neither of those seem like they're something that was due to the rush or could be justified by a rush or the lack of resources due to a pandemic. That's why I asked the question. But wait,
Jeanne, I think Jeanne wants to respond to that.
Good.
No, I was going to respond by saying that I think by, scientists don't just publish, they also read the literature. And I think that we watch the standard diminish over time. And I think that that really empowered some people to say, well, my work's no worse than this. And so I should be able to publish this. It's no longer something that I should wait and see and get more data for, because everyone else is doing it. So I'm going to do it too.
So with that said, and I want to draw on some conversations that we had together before this session, and I want to go back to that word stumbling, stumbling into, given, and Graham covered this a little bit in what he was saying earlier. Is there, the whistleblower, and if that's not the right word, forgive me, but the person that Graham referred to and that Jeanne referred to as being responsible for blowing the whistle on the hydroxychloroquine data, is there and I'm going to refer to that as the canonical whistleblowing regime, given what we have today in terms of publication technology, in terms of open source, and the full data sets being available, and the growing use of machine language. I did quick this is a passion of mine, the intersection of stem cell science and artificial intelligence, particularly machine learning and deep learning. Just before this session, I did a quick search on PubMed. There are over 500 articles in the intersection of those two technologies. How can we enlist the power of those technologies to address what you all have been talking about? Graham, I think you've got some thoughts on this, so let me turn back to you.
I just wanted to mention one more thing before I answer that question, Alan. And I think we talked about intent, we talked about stumbling. I think I'm going to repeat myself. We have to believe that most people do have good intentions, but there's a third class that we haven't actually talked about. And that's where someone believes so passionately that what they are doing is correct and is the way forward. They believe that the rules don't apply to them, that we would normally agree as scientists are the standards for publication. I am right. This data set maybe needs just a little massaging, or I just have to leave out this part, and then everybody will get on board. And when more data comes in with more patients, everybody will see that I'm right. We've seen that happen time and time again in regenerative medicine. Paolo Macchiarini, the absolute poster child of other people's rules don't apply because I'm right. Now, Alan, we are, I'll use your word stumbling again. We're still stumbling in scientific publications. We still have the same basic model as when I was a PhD student, that you went to the library, you took a tome down off the shelf, you opened it up, and there was the paper printed forevermore, carved out of stone.
Like the stone in back of you in the image. That's in back
right, and back when I was a graduate student, before there was paper.
Yeah. Papyrus. And but that's still the case, even though everybody's now reading the science electronically on a screen. We still use that same idea that this is canonical, that this is the paper, this is the only way it can be interpreted. I'm going to give you an example. When I first started publishing in stem cell research, it was a paper where we were trying to persuade blood stem cells to turn into brain. Okay, it was very, very popular at that time, was the idea that you could take any stem cell from the body and turn it into any other cell.
Lead to gold. Lead to gold.
Yeah, just by
the alchemist stream.
And the way, and Jeanne will smile knowingly about this, the way that we were demonstrating that our blood stem cells were becoming brain was by expression of something called nestin. And nestin at the time was seen as being a neural marker. Okay? We now know it isn't just a neural marker, it's an endothelial marker, it marks lots of things actually. But at the time, we knew, in inverted commas, that it was a neural marker, therefore we were turning blood into brain. Now, should that paper be retracted? No, it shouldn't be retracted.
No.
Because it was
No.
published and it was our best understanding of the knowledge. The data are correct. The discussion is hogwash because of what we've learned since. But the data are gold. The data should always stay the same. But we should now, in the modern age, be able to go back and put a note on that paper saying, by the way, we've learned a bit more about this since then. Sorry, boys and girls, it's not neural.
They do that with legal cases. I mean, Alan can speak about this, but Westlaw and Lexis, I mean, it's when something is not directly overturned, but you've got like a yellow flag on it so that, you know, as a lawyer, when you look at it, that it may not be good law anymore, but it wasn't directly overturned. I mean, there is a system to do that, or there's two companies that have done, three or actually have done that quite well. Perhaps we should start shepherdizing our
Beth, that's brilliant. Beth, that's a brilliant idea. That's a brilliant idea.
In this company, I will take that compliment. I will actually put it in stone somewhere.
So having said that, Graham and Jeanne, how do we do what Beth was just suggesting in science? Is it already there and we're just not seeing it, or should it be brought to the surface?
I'd like to see that, honestly. I mean, I think that means the author is taking responsibility for, and in Graham's case, it was really relying on someone else's report that particular molecule meant that it was a neuron. He wasn't the one who said that molecule meant it was a neuron. He was relying on someone else's paper, which, you know, if you keep going back, you realize that person probably should not have said that either. And so everything that he referenced that claimed that also should get a yellow flag. I mean, maybe not just two colors, but, you know, some kind of an asterisk kind of thing that this is a paper which stands on its own, but relying on the information at the time in retrospect is not, the conclusions are not correct. I mean, I would love to be able to do that. So because that we'd probably have half the papers ever published. Maybe 3/4 would have an asterisk, though. That's the problem.
So doesn't AI offer us a way of doing that?
I sort of do AI, as you know. So I'm machine learning anyway. I like it because it's unbiased, but I don't know how you can apply that to conclusions. Like what Graham described was really different from the fake data kind of publication. I mean, he's talking about wrong conclusions. We're talking in the hydroxychloroquine as paper as being deliberate, leaving out of data. I think there are some things in which AI or machine learning could help because it's an unbiased way of looking at things, but we'd need to train it so that it understood what we wanted it to do. And I don't see that quite clearly yet. Certainly plagiarism, that works for that. It may work for images so that you can look at an image on somebody's paper and say, that's the same image you published in a paper 10 years ago. But those are all like cheats, right? It doesn't do anything about the unintended mistakes that people make. Do you understand what I mean? So yeah,
sure.
It could probably it could probably remove a layer of problems, but it wouldn't remove all of them.
Yeah. Well, Jeanne, that goes back to what Graham was saying earlier about the concept of intent, doesn't it?
Yes, it does.
Graham, am I interpreting what you said earlier, Graham? I'm sorry,
yeah, I think you probably just get out of the game completely if you can't go in as an editor assuming good intent when someone puts. If you're looking at papers just saying, okay, I wonder what they've been up to this time, then Maybe you should become a policeman or something. I don't know.
Yeah, and that points to reputation is everything in science. Your history is what people, I mean, we're all people, right? We're all people as scientists as people. Our reputations are the most valuable asset that we have. And yeah.
It's your integrity. That's the end of the day.
Integrity.
All you have is your integrity, especially if you're
Right.
a clinician researcher. And you know what?
Yeah.
Nasty sidebar. You've got to be careful who you get in the sandpit with,
Yes.
or you'll catch fleas.
Yes.
And
That doesn't just apply to science.
No. And I have, and just
I've had many
as
a
an
conversation
example-
with Jeanne about this.
Yeah. No,
So
lots of
now-
things. But I was going to say, I have also done another thing. I have taken my name off the paper because I didn't want my paper to be in the list of authors, my name to be in the list of authors along with that individual. I mean, I just said, I know what the implications of this are, and I'm out of this.
And,
yeah, but
and if,
you're in a slightly different situation to say
I
that
know.
young researcher from a country that rhymes with Bina, who needs the publication in order to be able to move on with their career. And I get begging letters saying, I need this publication. And it's true, but it doesn't influence me in my decision-making process.
No.
But that's the big difference. Alan, to come back to your question about what we can use from AI, the obvious example I can think of, like we don't do it yet. We ask people to submit like the original Western blots and things like that, so that our reviewers can see what the images in the paper are based on. But we don't require people to submit their original data sets. But we could do that. And that
blockchain, it's a brilliant, I'm sorry, Graham, it's a brilliant application of blockchain, but keep going.
But you see, then that offers you the opportunity to aim technology at that data set. And on a very simple example I can think of is if someone is faking a data set, they tend to use random numbers, but that are in a certain range, that's the control group, and random numbers that are in a range for the experimental group. But real data don't look like that.
Yeah.
And people that are bigger jerks as me, we can look at a data set and tell you right away whether it's been faked or not because of that.
Yeah.
So that would be a very, very simple use of a computer's abilities. But I think what you would like, Alan, is to be able to use almost like a meta-analysis, to be able to use deep, high-powered learning to be able to look at multiple data sets.
Precisely.
That have something in common.
Precisely.
It'd be very, very difficult to do, especially in this field. But then that would reveal a deeper truth that isn't available to you
Precisely.
on paper.
Yeah.
So this would be
But who does that? Who does that?
That's exactly what I was just about to say. So what I'm grabbing from this is, and I'm sorry to Alan, because it's, I mean, there's obvious uses for AI and blockchain here, but either way, what I think we may think about is whether it's a third party of any sort that perhaps doesn't exist now, whether it's technology third party or like a Westlaw Lexis kind of third party who is sort of doing the indexing and the flagging and the link of the current paper to the other publications that call it into question or that supersede it or that have, used the science that's evolved, relying solely, and it's not that we shouldn't rely on the publishers and the scientists. We have to, but perhaps it's something that doesn't exist right now.
Yeah, so I'm mindful of the time, and there were some other things that we wanted to discuss as well. But to respond to what Beth was just saying, there is an organization called OpenAI, and OpenAI is responsible for a model. It's called GPT-3, Generative Pre-trained Transformer 3, which relies on a mathematical technique called autoregression. And it also has deep learning in it. And my suspicion is, and it would be really interesting to follow up on this, is that that kind of technology is what we would need. It's certainly not sufficient, but necessary for what it is that we're talking about. And we could probably spend the rest of the day talking about that. I could certainly, and I'm just mindful of the time. There were some other, the conversation up to this point has been very much what happened in the era of COVID. And what I want to turn to is the following. What was happening before COVID that because of COVID, we didn't have much time to focus on? So what was happening in October 2019 in the world of stem cells and regenerative medicine, which are pertinent to what we've been talking about, and how will those emerge in the coming months? And I've got two issues that we've discussed in mind. One is which Jeanne referenced in her introduction, which is engineering and manufacturing stem cells in a reduced gravity environment, and also, if time permits, the status of the so-called 14-day rule. So before we conclude, I just wanted to come back to that question about what will be reemerging as COVID hopefully winds down. So Jeanne, why don't we start with the International Space reference?
Yeah, right. So I am involved in a project in which we're sending neural organoids derived from iPS cells from people with Parkinson's disease and multiple sclerosis into space. Organoids, as many people give them a great deal of credit, but they aren't mini brains. They're essentially aggregates of neurons and other cells, if you add them, that interconnect with each other in a way that hopefully mimics some of the characteristics of those cells in the brain. So the idea is to, right now we're really in the discovery phase, although conclusions are coming out that are probably not going to be reproducible. One of the problems with space study on the ISS is that you can't send an experiment up every time you want to repeat it. You have to sort of like plan for it. So we have one data set so far from a rocket that went up in December of 2019 and came down in January in 2020, just before everything fell apart. So we've been spending the last year and a bit analyzing the data from that one experiment because everyone was shut down and everything started moving at a snail's pace. The idea, however, for doing things in low gravity, I think is it is in a little bit sort of analogous to other things that people have done with stem cells, which is let's grow them in low oxygen and see what happens. We're growing them in low gravity and see what happens. But it has relevance because it has relevance to space travel. Because if you have a little avatar of yourself as represented by neurons and microglia, for example, in an organoid made from your iPS cells up in space, then you can ask questions about whether it makes a difference whether you're in space or not. And now that we're discussing long-term space travel, I think sending avatars like that into space to ask very specific questions, like we're looking at neuroinflammation. We want to know whether it gets worse or better. But we're open to whatever happens so we're doing a broad analysis of everything about these organoids. We get to repeat the experiment, I hope, in August. So it will be, a very, very long time. Nobody ever does experiments over again on that sort of time schedule. But we have to do it twice. Otherwise, we don't think that we can publish the data. So there are lots of hopes about what can be done in low Earth gravity with stem cells. That maybe certain cell types that you can't grow on Earth you'll be able to grow in low Earth gravity. There are even hopes that maybe if you can build a model of a disease and send it up into space, that disease may get worse and look more like the later progression of that disease, and then test drugs on it in space. But everything has to be automated if you want to do it in space. So it's not the same kind of experiment. You don't say, postdoc, do that experiment again. You know, you have to plan ahead for it and have it, and nobody touches the cells, or nobody touches even the box that the cells are in for a month. So it's a very different way of looking. I think it's actually very valuable, because a lot of the things that we're learning about how to do these sorts of experiments, the automation, for example, that is required, the instrumentation that's required, the automatic microscopy, the sort of hands-off analysis of the progress of the cells, those are all going to be applicable to working with iPS cells on Earth, especially if you're doing this, the iPS cell, everything, but especially if you hope to use those iPS cells for clinical application, because it has to be reproducible, and having instrumentation that allows you to do a thorough analysis in an unbiased way is going to be critically important. But my motivation for sending cells into space is I'm curious about new stuff, as you guys all know. So the opportunity to find out what happens is always sort of been around in my entire life. This is one of those great opportunities, and I also really enjoyed the fruits of my labors, which was to go to Cape Canaveral and watch the rocket launch. So, you know, science is a multifaceted experience, and there's a lot more to it than doing experiments in the lab and writing about them.
So Jeanne, you just mentioned the use of organoids, and again in the prior conversations we've had, there's an interesting connection between organoids and the 14-day rule with respect to the development of human embryos. Let's take a few minutes, I think, to just explore that point. And again, why don't I let you begin, because I think you articulated that very nicely a few days ago when we were talking about this, and what that connection is, and the gastrulation concept. So take it on.
The first question, of course, is why 14 days? And that was an arbitrary number, an arbitrary timeline, because at 14 days during human development is when a process called gastrulation occurs. Before that, the embryo is pretty much amorphous. It doesn't have a lot going on in it. It's just a bunch of cells. But during gastrulation is when the cells start for their first time to do the processes that eventually lead to organs. Gastrulation is what the beginning of the formation of the brain is, really, really early. And so the concern was, should we, that a lot of people would think at that point that it was just a mass of cells the day before, but the day after, it is now something that has the potential of being a human being. And that concerns people. So extending that time means that if you could, in a culture or in a test tube or in an artificial uterus or something, make an embryo that goes beyond that stage. My question is, why would we do that? What can we learn? That answer has not been answered for me. I haven't come up with an answer myself. I'd really be interested in what Graham is thinking about, and also what Beth is thinking about. And in terms of, does the information that we can get from keeping the embryos longer, is it important enough to justify doing something that is going to raise a lot of concern among a lot of people, including scientists? Go ahead, Beth.
You can't put that on me. you know, if anything, I rarely express a personal opinion about any of especially these issues, and I won't. The only thing I actually just wanted to add something though, and please correct me if I'm wrong, all of you. I'm pretty sure that the original quota rule, we always refer to it as the 14-day rule, but I'm pretty sure that the language has always said you can, grow the cells to 14-day or until evidence of the beginnings of the primitive streak.
Yes, precisely, Beth. Yeah,
And that's the
shrinking
that's the
right.
beginning of gastrulation,
Yeah.
the primitive
Yeah,
streak.
Right,
But
and
it's a
the idea of the primitive streak, though, I think, to add it to that standard, is because it evidences perhaps some ability to either feel pain or be cognitive, or that was, I think, what the basis was. I'm not saying this is correct or not correct, but I think that the justification was that it might evidence the entity's ability to beginning to feel pain or to have some level of consciousness or whatnot. And I think that's probably, yes, is it or is it not an arbitrary day, 14 days? I won't come down on that, but I think it was for that.
Yeah.
I just
No,
you're
right.
I think that kind of puts a little bit of context as to, should we start to move the needle on what is potentially an arguable number of days to stop, and with the justification, which has always been the justification or some of the reasoning to use embryos at all, human embryos, which was, does the science outweigh the potential for all of the risks of using human embryos, which also includes, arguably, that we start to view it more as a commodity, and do we commodify human life? Now, again, I'm almost playing devil's advocate here, or God's advocate, that, whatever. But I do want to put that on there because there is, from the scientific end of things, it almost makes it a little bit easier than when you start to add some of the societal concerns.
Yeah. So I was just going to, let me give you an alternative to a beyond 14-day human embryo, and that's an organoid with neurons in it that can connect with each other and fire and communicate. What if I were to add to that the sensory neurons, so that could now take sensory input? You could drop a little bit of acid in the water and the thing would just freak out. Is that pain? Is that sensory? It's a little ball of cells that I made from iPS cells. Yes. And so, I mean, I think it.
No, I agree with you. And this is always one of my, the focus on human and humanness. I mean, it also goes to standards for chimera research. Are we making an animal too much like a human? I think arguably the standards that we've applied have always focused on humanness. If we were to talk about the pain and the ability to understand and feel suffering, we would have extremely different standards for science on animals. I mean, we don't even have to go to organoids. We can go to primates and dolphins and a whole classes of animals that can certainly feel pain and understand it. And so I don't necessarily disagree with you. I think you're making an excellent point, and there's more of them, more analogies to be made there.
Graham, with respect to all of that references, all of the references there to organoids and the comparison to embryonic stem cells, and from the publication standpoint, any thoughts?
I think, first of all, I don't think anybody's actually said this, but I think it has to be clarified that when that 14-day rule was thought up, it really wasn't because of what happens at 14 days. It's what everybody had a shared understanding wasn't yet happening at 14 days.
Mm-hmm. Right.
Nobody would get squeamish at 14 days. Let's set aside what it is you're actually looking at in terms of source material. It's what wasn't happening. And we had no clue how to allow it to develop for 14 days when those decisions were made. This has all been accelerated as a discussion by that being achieved at the University of Cambridge. They now are capable of taking it out for 14 days.
Yep.
And in their wisdom, societies and people are now publishing opinions on this. Are they talking to Joe Public about it? Apparently not. There has been no consultation outside of the scientific community to actually have a proper conversation about how people judge this work, it's all scientists talking to each other. I mean, have we learned nothing about how to be received by the public, that we're making these mistakes again and again? And then
I take slight exception to that, as having led the New York State Stem Cell, the Ethics Commission, as well as the whole board, on its decision to use embryos in research. And it was actually amongst this is why I'm so sensitive about, expressing personal opinion and whatnot, because to lead these discussions, you're not there to expert, but to conduct these discussions with people who do believe this is murder. These are the toughest discussions to have, and to respect, various viewpoints in this realm is extremely tough. I agree, Graham, that there's certainly not enough of that discussion being had, and certainly not having multiple stakeholders. I mean, unfortunately, we don't do this with very much of anything, but to have multiple stakeholders at a table and actually try to come to some sort of an understanding, even if we're going to agree to disagree, but to have some discussion that is respectful and attempts to reconcile what are irreconcilable differences.
No, and
Yeah,
I applaud that, Beth. I really do. But I think you did concede, though. It's the exception what you were involved with.
Oh yes, this is why I love that job. It does not exist usually.
And then we're going to be surprised when Capitol Hill says, "You guys have just started doing this. You've decided this. You've decided this is okay now." But the arrogance is absolutely joking.
Yeah, you have to be arrogant to be a good scientist, though, honestly. But Graham, what do you suggest would be the best way to conduct a conversation with people other than each other? If we wanted to open up the discussion, I mean, is this Twitter feed stuff?
No.
Or what?
Okay, so let me give you an example. So most people don't seem to realize this, but I am still a scientist at a university.
I knew that.
and we do environmental science, but we don't do it in a vacuum. We have a community advisory board with 40 stakeholders from various organizations in Detroit. I meet with them 3 times a year. They review our applications for pilot projects. If the community doesn't agree that a piece of work is worthy and should be going forward, it doesn't get funded by us.
Mm-hmm. But the NIH doesn't do that.
It's insane. The NIHS does it.
Mm-hmm.
So,
but when you don't have, I think, a very different realm than regenerative medicine and stem cell research, I think it's pretty obvious from the pandemic alone, is that when you don't have community buy-in and you're not taking viewpoints and the understanding of science, or where the science has come from, or the justification of it, what happens when you just try to launch it out into a community without having any sensitivity or any understanding of what it's going to mean or what it looks like. It has extremely bad ramifications for respect for science and for the communities whose experiences and viewpoints haven't been taken into account. You're actually arguably putting them at greater harm, because they're not going to accept, understandably, healthcare and science advances, because they don't know where it comes from, and there's not been any engagement to have anybody trust it.
I want to thank all of the people in the audience who are watching this session. We could spend the rest of the day talking about these topics, and I'm not sure that we would keep your attention for that long. So we certainly want to thank all of you, and I certainly want to thank all of my co-panelists, Jeanne, Graham, Beth. I want to thank you. Doing this each year with the 3 of you is a tremendous learning experience for me. So thank you.
Thank you, Alan. That's great.
Thank you for being our very thoughtful, fearless leader, and contributing substantively, and just in your leadership and thoughtfulness. It's always, this is always my favorite panel of the year with the three of you. And I'm very lucky to be part of it.
Thank you, Beth. Thank you for saying that.
Okay, goodbye, everyone.
Bye.
Thanks. Bye.